Now let's start with the third most important activation part or activation pathway of the complement system, that is mannose binding lectin pathway. So it is mannose binding lectin, it is MBL pathway. So basically in our body, lectin is a particular protein which is produced in trace amounts. Now the lectin is actually a very important protein because it has to attach to those cells which try to or which have exposed mannose on them or specific sugars on them. Now our normal cells do not have these sugar residues like mannose, but the bacterial cells they do have. The bacterial spores, even the fungus and even the yeast, they have that. For example, if I talk about bacteria, so there are particular examples here like bacteria including Salmonella, Listeria, then fungi including Candida, and then yeast including Cryptococcus. So, sorry, yes. So, bacteria can include Salmonella, then Listeria, the two of them, Listeria L. Then there are fungi, and then there are yeast. Fungi include Candida, and then there are some yeast like Cryptococcus. So the cell walls of these bacteria, fungi and yeast, these organisms particularly present mannose on them. So whenever such bacterial cells or fungal cells or yeast cells are inside our body, the mannose will be present on them. So that time this lectin will bind to that mannose. And this lectin which is binding, suppose this is mannose and this is lectin. So this lectin and mannose is very similar to C1q complex. So here this mannose and lectin, when they bind together, they will be very similar to C1q complex. After that they will start their activity. So they will activate MBL associated serine protease, that is MASP1 and MASP2. So once the mannose and lectin have bind together, this ML complex or MBL complex, this MBL protein, mannose binding lectin protein, which is very functionally similar to C1q, it will activate the two further proteins, that is MBL associated serine protease type 1 and MBL associated serine protease type 2. This will functionally resemble C1R, this will functionally resemble C1S. So if you remember, we had C1 including C1Q R2 S2. So the similar is with here also. The Q is actually MBL itself, R is MASP1 and this S is MASP2. When the three of them are activated, these now then start their pathway very similar to the classical pathway. They will now activate C4. This C4 will then the C4b will bind to them. This will further activate C2, then this will activate C3, and this whole of the complex will activate C5, means they will be called as C5 convertase, the C5a acting as NF-ilotoxin, and then C5b, this C5b then forming 6, 7, 8, 9 together forming the membrane attacking complex. So very similar pathways then followed. So there are some initiating points which are different in all the three pathways. More or less the another whole part is very similar. So this is what you need to know in the mannose binding lectin pathway, which is one of the activation pathways of the complement system. So here are the three major pathways which we have discussed about the complement system. And if you want to know about the structural details also of this, so here we have the structure of C1 macromolecular complex. We already knew that C1 consists of Q, R and S subunit. So here you see this is represented as C1 Q R2 S2. So there is a C1q molecule. It consists of 18 polypeptide chains arranged into 6 triplets. So this whole stock is made up of C1q. So you don't need to learn the numbers, but remember the stock, which consists of many polypeptide chains, are forming the C1q part, and each of them contains one A, one B, and one C chain. That is again if you need to know and if you need to learn. Further on above this, there are the parts where we have C1R, C1S. These are also bind to this. So the stock is made up by C1q and the heads are made up by C1R and C1S. So that is the whole part which we were talking about, the structural detail and then the complement system and the activation pathways.